She started her research career as a graduate student, being interested in cancer and signaling mechanisms that contribute to cancer development. Then for her postdoc, she moved to the Man lab, where she mastered the application of mass spectrometry to analyzing proteins. And she returned to Israel to establish her lab about a decade ago now, where she has done a lot of interesting studies applying mass spectrometry to understanding cancer biology, and a particularly interesting recent work that she has done, still using bulk approaches, is to understand the links between immunometabolism and mitochondria and cancer progression. And
so for some reason, it doesn't show what the present.
Let me see. So you're a co-host, and that means you should be able to share your screen. Do you see the green button at the bottom?
Yeah, I know. I'm clicking on it, but it doesn't show. But sorry for that. So let's start with that. And
yeah, now it's working.
Yeah, no, I'm sharing, but not the right thing. Okay, so we'll get there. Sorry for that. Can you see that?
Yeah, now it's working.
Okay, so thank you, Nikolai, for the invitation and for scheduling me in the beginning so I can actually fit still the daytime here in Israel. And I want to talk about our clinical cancer research work, and a bit about our single cell proteomics work that we have been doing in the lab. But I want to first start with a kind of our motivation to do what we are doing. And just a few facts. So there are 40 million new cancer cases per year worldwide. 38% of us are going to be diagnosed with cancer. And the quite amazing fact is that in the last 10 years, there was only 1.5% reduction in cancer mortality. And what are the treatment options? So surgery and chemotherapy and radiation are probably the oldest treatment. And then moving on to targeted therapy, and the most modern type of therapy is immunotherapy. But despite having all of these options, we're still having this very grim reality. And this is associated with very critical questions that we're trying to answer in the lab. So how diverse are the tumors? How dynamic are they? Why can't we diagnose cancer early enough? And why don't we cure all cancers? And the answer to most of this lies in the heterogeneity and the diversity of human cancer. So obviously we all know that there are different cancer types. There is melanoma, breast cancer, lung cancer, and so on, but there is also high diversity between the different patients. There is, on top of that, there is high diversity between regions. So if this is an H&E staining and you take sample different regions, there is high diversity between them. And obviously there is also diversity between the cells. So each region can be composed of different cell populations that have different between each other. And there is enormous amount of work trying to understand this heterogeneity on the molecular level. While obviously most of the work that is done on the genomic and transcriptomic levels, but I probably don't need to say this to this audience that proteins are much closer to the function, and therefore we want to look at the proteins and the protein and the heterogeneity on the proteomic level rather than transcriptomic and genomic. But on top of the obvious, there is also one important aspect to it, that when we talk about cancer, there are a lot of stochastic events, mutations, chromosomal aberrations that happen on the genomic level. So the heterogeneity between cells is much larger on the genomic level, and this is reduced when going to transcripts, and even more reduced when going to proteins, and therefore it is actually much easier to understand the biology when looking at the proteomic level. And in today's talk, I want to talk about this. I will talk about these 3 layers of diversity: diversity between patients. I'll talk about breast cancer classification, diversity between regions. I'll talk about the intratumor heterogeneity, and I'll talk about our development of the single-cell proteomic pipeline towards understanding of melanoma response to immunotherapy. So starting with breast cancer, a bit of an introduction. On the RNA level, people have been, there are 4 main breast cancer subtypes that are very well known in the last 20 years or so. The luminal A and luminal B, these are hormone positive, ER positive, estrogen receptor and progesterone receptor positive. While luminal B is more proliferative, has high Ki67 staining. HER2 enriched has amplification of the HER2 gene, and basal-like, which are mostly triple negative, don't have expression or low expression of the ER, PR, and HER2 receptors. Now, this is a figure from one of the largest breast cancer genomic analysis of the TCGA, published in 2012, and they associated point mutations with each of these 4 subtypes and associated them with the clinical data and with copy number aberrations of these tumors. A couple of years ago, Gali in my lab took the proteomic approach, and she assembled a very large cohort, actually the largest breast cancer cohort at the time, with more than 200 samples. And what we wanted to see, after validating the known differences between the subtypes, we wanted to see whether we can actually challenge the existing classification of these 4 subtypes. And Gali performed consensus clustering analysis of these breast cancer samples, and she found also 4 subtypes, but when she compared the CC, the consensus clusters, to the clinical classification, she actually saw that this is not exactly the same. So what she found is that we actually have 3 clusters that are estrogen receptor positive or luminal subtypes. The HER2 subtypes is actually not forming one coherent subtype, but actually kind of mixed all over. And the triple negative subtype forms a distinct cluster. She could also see that CC1, consensus cluster 1, is luminal B, but eventually she saw that CC2 and CC3 are both luminal A clusters. And as a reminder, on the RNA, we only see 1 cluster like this in many, many analyses, many papers. We could also validate our results using RPPA, so another approach of protein analysis. And what this comparison showed us is also some clinical relevance of this novel luminal A subtype, because it showed us that it may be associated with tamoxifen, with treatment resistance of luminal A tumors. What was interesting is when we compared the CC2 and CC3, the 2 luminal A clusters, we could see hundreds of proteins that differentiate between these 2. But when we compared this to RNA, we took these proteins and some, or genes, and we actually saw a total mess. We couldn't see any association with different clusters, which means that we actually don't see these 2 subtypes at all on the RNA level. So the protein-based classification identified a novel cancer subtype with potential clinical relevance, which is not seen on the RNA level, and this is kind of reinforces the significance of analyzing the protein level. However, we're left with critical clinical questions. So upon proper classification, why don't tumors vanish upon treatment? So even why don't we, when we treat with tamoxifen, why don't the tumor eliminate completely? Why don't tumors respond to targeting or driver mutations in many of the cases? And why do metastases very often differ from the primary tumor of origin? And the answer to all of these questions lies in the internal heterogeneity of the tumors. And here I'm showing you an H&E slide of a tumor from one of our cohorts. And we stained these tumors for the receptors, HER2, progesterone receptor, and estrogen receptor that I don't show you here. And if I show you here, this is the region taken from here. very strong staining of HER2, no staining at all of the PR. I look at a different region, only a few millimeters apart, less than 1 centimeter away, very low staining of HER2 and very strong staining of PR, and a third region with no staining of HER2 and very sparse staining of PR. And this is really just within a single tumor, a single part of a tumor, few millimeters away from each other. And Maria in my lab took the project upon herself to basically characterize from scratch the heterogeneity of the tumors. And the idea was to take the tumors, take the entire tumor, slice, stain, and map histopathologically all the entire tumor, then laser capture, microdissect each region, and pool samples using TMT and run on the QX-active HF. All together in this project, we analyzed 330 tumor regions coming from 35 patients. So also we include 35 normal samples. We also have 50 lymph node metastases and 250 stromal regions that we haven't analyzed yet. And overall, looking at all of these data, this is just the tumor, so not yet the proteomics data. On the upper part, you can see the molecular subtypes, so estrogen receptor positive, ER/PR positive, PR, HER2, triple negative, and triple positive. The bottom part, you can see all the patients. Already from this picture, you can appreciate the extent of heterogeneity that exists just from the mapping of all of these tumors. You can see that most of the tumors actually come from different subtypes. And only few tumors, mainly coming from triple negative, are more homogeneous. For example, this one or this one here. Now, one technical advantage is that we get from analyzing these regions is that we get very high statistical significance. So if we compare here, for example, hormone positive versus HER2, or hormone or versus triple negative, or triple negative versus HER2, if we kind of average all the regions in a form of pseudobulk, we get very few significantly changing proteins. While when we analyze the regions, we get hundreds and even thousands of significantly changing proteins between the regions with these characteristics. So we get much fewer populations, which give us much higher, much stronger statistical significance. But what do we see from all of that? So when we look at the proteomic data and we cluster the proteomic, all the proteomic data, what you see here, every row here is a different region. We can see very clear separation between the normal, which are marked here in red, and all the rest of the cancer regions. We can also see separation between the patients. So each color here is a different patient, and we can see that in most cases they're clustered together. And we can also see that the separation into a bigger cluster makes a lot of sense. We see here clustering of the triple negative, we see clustering of ER/PR, triple positive, and so on. However, we can also see a lot of outliers. So for example, there are two examples here of a single patient. So here, which has a single patient that has ER/PR regions as well as triple negative regions, which are clustered very separated from one another, although they're coming from the same tumor. And here another example of separate ER/PR and triple positive. They are very different from one another. And a very interesting aspect that we saw in our data is that, for example, in these cases, we have the triple negative regions, but we also see many triple negative regions that are embedded within other tumor types. And actually what we realized is that these triple negative regions here are taken from very homogeneous tumors, while the triple negatives all around are taken from, so these are from homogeneous tumors and these are from heterogeneous tumors. And what we could actually see is a very significant difference between homogeneous and heterogeneous tumors. And what we are studying now is trying to understand why this heterogeneity is, why are these differences caused, and what are the molecular mechanisms that are associated with these differences. So the analysis of the regions showed us that large tumors are composed of multiple histological and molecular subtypes. The internal heterogeneity affects cancer classification and treatment response. And the topological proteomic analysis identified significant proteomic differences between homogeneous and heterogeneous tumors. And we're now looking at the involvement of the immune system on in eliciting this tumor diversity. And now the question is whether the homogeneous regions are actually homogeneous. So when we took and isolated these regions and we assumed that they're relatively homogeneous, we actually know that they're not perfectly homogeneous. Probably there are additional subpopulations within. And we want to see how this heterogeneity is reflected on the single cell level. And while in this study, we focused on the cancer cells and we kind of eliminated all the immune cells from the analysis, it is very intriguing to see how the immune system is affected or affecting tumor heterogeneity on the single cell analysis level. I probably don't need to explain the importance of or the comparison between bulk versus single cell. So obviously in bulk, we have bulk analysis. We have the advantage of getting deeper into the proteome and we see the main cell population, but what we are missing is really understanding all the cell populations. And this is something that we need to go for single cell analysis. And what we have been working in the lab, and this is led by Lir Beck in my lab, a PhD student, we're looking at three aspects. So one aspect that I'll tell you about is the microscale sample preparation that we're working on, implementation of the scope MS, and analysis of tumor cell populations, kind of going more to the biology in parallel to all of this, and we're trying to move to the three directions more or less in parallel. Okay, so kind of moving to the microscale sample preparation, we really love a methodology that was developed in Jeroen Kruisveld's lab, which is called SP3, single pot solid phase enhanced sample preparation. This is based on carboxylated magnetic beads, and this allows lysis, cleanup, digestion, labeling, and peptide cleanup, all of that on the beads. We use it also for a lot of our clinical, bulk clinical work. And since this really gives us very sensitive sample preparation, we also decided to implement this or try to implement this for the single cell. Jeroen actually presented this in the single cell meeting in Vienna last year. And he also showed automation of this system, and we actually did in parallel relatively similar protocol. We fully automated the SP3 on the Bravo liquid handler of Agilent, and this allows us to work in 96, and we recently moved also to 384 well plates. And this allows really robust analysis and very efficient analysis of very, very low sample amounts. And this is just to show moving from 1,000 cells that we identified more than 2,000 proteins to 500, and we reach almost 1,500 proteins from only 200 cells. So we really get very robust, reproducible, and sensitive sample preparation. And eventually the samples are rather clean, which allows us to safely put them into the mass spec. Now our idea was to take the SP3 that we really like and combine it with the scope MS. And the idea, and I guess I don't need much introduction to that. And basically the idea is that we have, in this case, TMT 10-plex. Each cell is labeled with one TMT channel. Then we have an empty channel where we have a carrier of 200 cells. We played a bit with the number of cells, but eventually most of what we do is with either 100 or 200 carrier cells, and we analyze all of that on the QX-active HF, and analyze all the data with MUX1. Thank you for developing this. It's really helpful and works really nicely in our hands as well. And the idea here is basically combining the SP3 with SCOPE-MS to create one coherent workflow, starting with protein extraction, binding protein to beads with ethanol, cleanup, trypsin digestion, then TMT labeling and multiplexing, and then binding the beads, the peptides to the beads with acetonitrile, cleanup, and releasing the peptides with DMSO. So that really works very nicely and smooth. And then from the robot, basically it goes directly to the mass spec. And this is just our first, or one of the kind of tests on analyzing 10 cells with a carrier channel of 1,000. So this is kind of a safe test. We can see overall more than 1,400 cells, and we continuously work to improve the methodology. So this is, for example, comparison between 250 milliseconds and 500 milliseconds injection time, and you can see that 500 increases the number of identification and obviously the quality of the MSM spectra. And using similar workflows, we can really very nicely separate between two cell types, so Hela and cervical cancer cell line and a melanoma cell line. And this is rather routine and simple. Moving to the single cell, we use here a single channel of 200 carrier cells. And again, we can see, I would say up to around 500 or so cells, proteins per cell. And when we use two carrier channels of 200 cells each, we can reach more than 800 proteins with a 500 milliseconds injection time. So we see the benefit of two carrier channels and longer injection times is increasing the overall protein identification. Now, in parallel to implementing and developing and improving the single cell approach, we also want to go to the biology and kind of combine all of these approaches. And our main interest for this project is looking at the immunotherapy response. And I just want to kind of give a brief introduction. So basically, we're studying immunotherapy response, and we started this research a few years ago, and the analysis of the bulk sample was already published more than a year ago in Cell. The idea of immunotherapy in general, for those of you who don't know, is to strengthen the immune system to target the cancer cells. So basically, our immune system is fighting against cancer on a daily basis or an hourly basis. And the cancer cells in parallel develop ways to inhibit that. What immunotherapy does is basically to strengthen the immune system. The most known or used types of immunotherapy are checkpoint inhibitors, so basically antibodies that inhibit the inhibition of the cancer cells on the immune system. And another approach is based on adoptive cell therapy, so here in the form of tumor infiltrating lymphocytes. So basically T cells are extracted from the tumors, expanded and activated ex vivo, and then reinfused back into the patient. And what we did in this study, and this was led by Michal Areli in my lab in a close collaboration with Gal Malkel from the Sheba Medical Center, and we analyzed 116 tumor samples, all bulk analysis, from TIL treated and anti-PD-1 treated, from responders and non-responder. Everything was pre-treatment. Basically, it might be difficult for you to see, but looking at all of these clinical parameters, the doctors are never able to predict who is going to respond or not. despite the fact that there are huge differences in overall survival and progression-free survival between the responders and non-responder. And making a very long story short, using this bulk analysis, we're able to extract signatures of response, one signature of the TIL treated, one signature of the anti-PD-1 treated, and interestingly, there was absolutely no overlap between the signatures. The TIL signature highlighted metabolic processes, mainly lipid metabolism processes. The anti-PD-1 signature highlighted interferon gamma related processes and antigen presentation proteins, which were also seen on RNA, parallel RNA studies. But actually the metabolic signature was totally unique. Nobody knew about this association with response. So it was interesting that the signatures were totally different, but when we looked at the functionalities, we actually saw that they're rather similar. And when we combined the data sets, we saw that the immune response, immune-related proteins create a very dense network of proteins together with the metabolic proteins. So they're both relatively high in response, or significantly high in responders to both treatments, TIL and anti-PD-1 treatment. And we went on and we were able to actually perturb these pathways. And for example, here we knocked out ACAT1, one of the key proteins in the TIL signature, which was associated with better response. And this led to increased tumor growth in vivo, which was caused by reduced antigen presentation. So the tumors are less visible to the immune system, and therefore they grow much faster in mouse models. And we're now trying to understand this better, the biology of it, and trying to also activate the lipid metabolism in order to increase immunotherapy response also in patients. So moving from this bulk analysis forward, we are asking ourselves several questions like, what are the proteomic differences between responders and non-responders on the single cell level? So in that project, we focused only on the melanoma cells, and we kind of ignored everything that is happening to, for example, the T cells, which are really critical in this aspect. How do cell populations change upon treatment? So we want to see the patients before treatment, but we also want to see what is happening right after treatment. And how general is the association between immune and metabolic activity? So what we are doing nowadays, and I don't have any results to show you yet, but this is kind of ongoing, is to take patient samples before treatment and on treatment, then isolate, separate the cells, separate between the melanoma cells and all the stromal and immune cells, sort them directly into lysis buffer, and then run it on the mass spec. And until this is actually happening, and we're kind of limited by the availability of clinical samples, we're doing some mouse work in a melanoma mouse model. And for now, what I want to show, just very kind of preliminary results, that we are sorting pools of 1,000 cells from 13 cell populations. So very sophisticated sorting panels of myeloid cells, lymphoid cells, and cancer and stromal cells, altogether 13 populations. And very preliminary results show that in a label-free analysis of the melanoma cell populations, we are able to identify more than 1,000 proteins for almost all cell populations that you can see here. Even from very rare cell populations such as NKT cells, we reached around 800 proteins from 1,000 cells. And we could also see very known cell type markers of each of these different cell populations. And we can very nicely also see that the cells from the same lineage are co-clustered, and even from 1,000 proteins per cell, per cell population, and a total of 2,500 cells, 2,500 proteins, we can actually make a lot of biological sense. And currently, actually, the full experiment is being analyzed in the mass spec, and I hope to be able to share it with you next year when we actually meet in person, hopefully. So I want to summarize, what I showed you is that analysis of breast cancer heterogeneity unravels the functional significance of heterogeneity itself. Moving to the single cell or the minimal amount analysis, we automated the SP3 protocol, and we identified thousands of proteins from pooled cells, and implemented the SP3 scope MS and identify hundreds of proteins per cell. And with the pooled cell populations, we can unravel cell-specific markers and processes, and we are really moving on to analyze the biology, where we are going to apply the single cell SP3 scope MS to tumor cell populations to unravel the dynamics of cell populations in an unbiased manner. And with this, I want to finish and thank all the people in my lab who really work very hard to do this work. And I mentioned all of these people during the talk. I also want to thank my clinical collaborators that are really critical in all our analysis. I want to thank you for the attention and for joining, and I hope to meet you all in person next year, and I'm happy to take questions.